So we have situated ourselves firmly within the muscle. We've done a deep dive from the muscle belly into the cell, into the muscle fiber. Right? And we talked about all the structures within the muscle fiber and how they're organized, and the myofibrils being those that we're really interested in, because that's where the filaments live, myosin and actin, and that's where contraction occurs. And we introduced the idea of how the signal is sent to the muscle last class. How the signal is sent to the muscle. We are going to connect that signal to contraction today. How does that signal result in contraction or shortening of the muscle? Now, I introduced the idea of shortening by talking about myosin and then pointing out that it's not compression of the muscle. Instead, muscle contracts because those filaments will slide over top of each other. Actin and myosin that have an overlap will get more of an overlap. They slide together. So the Z discs, the bookends of the sarcomeres, move closer together. And with multiple sarcomeres set up in sequence, the overall effect is the whole muscle shortens. It doesn't compress, but the filaments slide together. The sarcomeres get shorter. Then, even though we're only really focused on actin and myosin from their active standpoint, they are involved in actively shortening the muscle. The structural proteins that we talked about help make sure that force is transmitted the entire length of the fiber. We'll see how titin comes into play in the video today. And we mentioned dystrophin, which are the anchors at the ends of the myofibrils. Within the muscle cell, the entire myofibril is anchored by dystrophin at the ends. So before we see it in action, let's get a sense of how the sarcomere changes during contraction. How the sarcomere changes. So this is the classic example that I like to use, the elbow flexion or biceps curl. Almost fully extended at the top, and then progressively more flexed as you move down through the figure. And so we're looking at muscle shortening. As you move down, the muscle shortens, right? The muscle contracts. Notice the structures, the compartments that we identified within the sarcomere, how they change. The H zone and the I band, remember those were named for the width of the letters. The H was slightly wider, contains only myosin. I is a thinner letter, contains only the thin filaments, actin. As those fibers, as those filaments gradually overlap more and more, as they slide over top of each other, those areas compress. The H zone and the I band are smaller because there's more overlap and fewer sections, less sections of just those individual filaments. You can see that on the right-hand side as well if I highlight them for you. The H zone and the I band. As the muscle shortens, they get smaller. But notice the size of the filaments doesn't change. Myosin is still the same length. Actin is still the same length. Right? The red proteins, the yellow proteins, they're all the same length. There's just more overlap. The lengths are unchanged, even though everything seems to be compressing. Filaments slide over top of each other, hence the name, the sliding filament mechanism. So let's look at that in action. Let's look at a nice animation that shows you how myosin walks up or climbs the actin rope.
An action potential propagates along a single motor neuron from the brain or spinal cord to the end of the motor neuron near the skeletal muscle cell. This triggers the events at the neuromuscular junction that result in an action potential arising propagating along the muscle cell membrane, or sarcolemma. As the impulse moves down the sarcolemma, it continues to travel deeper into the muscle cells through membranous structures called transverse tubules. The interior of the skeletal muscle cell is filled with rod-like structures called myofibrils. Each myofibril is formed by repeating structures called sarcomeres, which are the muscle cell's contracting unit. Each myofibril is covered by smooth endoplasmic reticulum, called the sarcoplasmic reticulum, which stores calcium ions. Sections of sarcoplasmic reticulum are located along either side of the transverse tubules. As the action potential travels down the transverse tubules, voltage-dependent calcium channels in the sarcoplasmic reticulum open and release calcium into the sarcomeres. The calcium activates the sarcomere to contract. Each sarcomere is made up of three types of filaments: tightened filaments and thin and thick filaments. The tightened filament acts to provide elasticity for the sarcomere. Thick filaments are surrounded on each end by 6 thin filaments. When the sarcomere contracts, the thin filaments slide over the thick filaments by a process known as the sliding filament theory. Thick filaments are composed of 2 myosin filaments twisted around each other, and which are anchored in the center of the sarcomere. Each myosin has a head and tail. There are binding sites for ATP and for actin on the head of the filament. Energy in the form of ATP moves the head into an activated position, much like cocking a gun. Once bound to actin in what is known as a cross bridge, the myosin head will bend and move the actin. The thin filament anchored to the ends of the sarcomere is primarily composed of actin proteins. Each actin protein has a binding site for myosin. The binding sites on actin proteins are covered by tropomyosin, which is maintained in place by troponin. Troponin is a protein spaced along the thin filament and attached to both actin and tropomyosin. It also has a binding site for calcium ions. In the absence of calcium, the myosin filaments are prevented from binding to actin filaments because the binding site on actin for myosin is covered by tropomyosin. However, when the sarcoplasmic reticulum is stimulated, it releases calcium into the sarcomeres. The calcium ions then bind to troponin. Troponin then changes shape and pulls the tropomyosin to the side, allowing myosin to attach to actin. When myosin forms a cross bridge with actin, then the myosin head bends as it slides actin toward the center of the sarcomere. Contraction of the sarcomere is a series of stepwise cycles occurring between the thin and thick filaments simultaneously throughout the sarcomere.
So the first part of that video looked like a lot of review, right? And you thought it was going to be boring, and then it just ramped right up. We introduced the regulatory proteins. We'll come back to those in a little bit, or actually on the next slide. But importantly, you saw the sliding filament mechanism, or the sliding filament theory as the video called it. Myosin moves actin, but only when calcium is present. So that's the point of connection, right? The action potential triggers calcium release. Calcium allows myosin to bind to actin. The presence of calcium allows the muscle to contract. That's how excitation is coupled to contraction. You'll often hear that phrase, excitation, contraction, coupling. Excitation, contraction, coupling. The muscle can be turned on, but because calcium is released, being turned on results in contraction. So, the sliding filament mechanism, all those players that we just saw in the video are shown here. It's a good exercise for you to try to identify each one and figure out the sequence as we go through. But basically, as soon as the action potential is delivered, you have this animation in the slides on Moodle, so you can just, if you put it in full screen, it'll run for you. As soon as calcium is released, it binds to troponin and moves tropomyosin out of the way, or troponin. Troponin, troponin. It binds and moves it out of the way, and by being out of the way, the binding site for myosin is now available. It's available. So if it's available, myosin will bind it. In order for there to be shortening, muscle, in order to contract, needs energy. And so myosin will break apart ATP. That's that big dark green sphere that gets broken into two pieces. ATP, we talked about last class, is the energy currency of the cell, and we break it apart to get energy to do work. Myosin breaks it apart to get energy to contract the muscle. And in doing so, powerfully contracts and moves actin, shortens the muscle, shortens the sarcomere in what's called the power stroke. So ATP breaks apart, gives myosin the energy it needs, and the myosin powerfully contracts, cocks its head to pull the actin filament. We'll take a look at all the steps involved in that sequence of events coming up. When the energy is consumed, ATP is released, or whatever the products are, the breakdown products, they're released. Myosin resets, troponin resets. As calcium gets taken out of the cell, it resets, and then the binding site's blocked again. Only when calcium is present is the binding site available. Only when there's energy available does myosin contract. Both of those things allow shortening of the muscle fiber. So calcium needs to be there. Energy needs to be there. Let's look at the steps in the contraction cycle. If we assume that calcium's been released, what are the steps that myosin takes in order to pull on the actin filament? This is a series of events in a cycle that's always repeating. It repeats the entire time you're contracting the muscle. It tries to repeat when you relax the muscle, but even just sitting at rest, there's constant tone in the muscles. It's always going on. The first thing that happens in a fresh myosin molecule at rest is it searches somewhere in the cell and looks for energy. When it finds that energy in the form of ATP, it binds it, and by binding ATP, it reaches. That's the cocking the gun analogy that was in the video. By binding the energy, it reaches forward. You can see that reaching here. This is a 90-degree angle. I just put that angle up here so we can compare the difference in reach to the second phase of myosin. You can see the head is placed at 90 degrees, which allows it to reach out and then across to bind actin if it's available. Myosin grabs ATP and reaches. In reaching, it breaks the energy up. That's what the word hydrolyzes means. Hydrolyzes means to break up using water. Hydro, water, lysis, break apart. If calcium's present, then myosin reached for good reason. If calcium is present, that means that troponin has moved out of the way, tropomyosin has revealed the binding site on actin for myosin, and a cross bridge can form. Cross bridge is the name of the situation when those two molecules are connected, when they're bound. Cross bridges can only form when calcium is present. So this is the energized pre-contraction state. Myosin has the energy on hand. It's bound to actin. And now it needs to do something with that energy. Oddly enough, doing something involves the breakdown products of ATP detaching. And as they detach, that prompts myosin to pull. ATP is adenosine triphosphate. When we break off one phosphate, we're left with adenosine diphosphate, so ADP, and this is just inorganic phosphate, one phosphate molecule. Those break off, leave myosin, and by doing that, it pulls actin close. You can see the difference in the orientation on the bottom panel. Instead of sitting at 90 degrees, the head has... Is that alarm to wake up? Yeah? The head of myosin now sits at 45 degrees. It's pulled actin by releasing ATP. Well, as soon as that's done, contraction in this phase is over. This myosin molecule has contracted. It can't do anything else. It needs to reset. And so it resets by letting go of actin and then looking for the next ATP, the next energy molecule that's circulating around that allows it to reach and repeat the whole process over again. So myosin binds energy and it reaches using that energy. It grabs onto actin if calcium lets it. When the byproducts are released, myosin pulls, it contracts, and then there's nothing left to do but let go. It lets go of actin until the next ATP molecule is bound. These are the 4 steps of the contraction cycle. Every single myosin molecule does this. They don't do it necessarily all at the same time. There are hundreds or thousands of myosin proteins in the muscle at once. But if you zoomed in on one and looked only at one, this would be the cycle that it would go through. When you add them all up, we get full, complete shortening of the muscle. Let's add them all up and see what that looks like.
The first step of the contraction cycle occurs when ATP, already bound to the myosin head, is hydrolyzed. This provides energy to position the myosin head in its cocked position. Next, when calcium ions are released from the sarcoplasmic reticulum and attached to troponin, it uncovers the binding sites on actin. Myosin forms a cross bridge with actin. Once this occurs, the myosin head bends from its cocked position and slides the thin filament towards the center of the sarcomere. This process is referred to as the power stroke. In the final step for each contraction cycle, ATP binds the myosin head, resulting in detachment of cross bridges. As long as the levels of calcium and ATP inside the cell are adequate, then the contraction cycles will repeatedly occur and the sarcomere will shorten. That is, as long as the skeletal muscle contains enough energy and is stimulated by nervous impulses, a continual shortening of the sarcomere will occur. Calcium pumps in the sarcoplasmic reticulum pull the calcium ions out of the sarcomeres back into storage. When the calcium ions are removed from troponin, it returns to its original configuration allowing tropomyosin to again cover the binding sites on actin. Myosin can no longer attach to actin, and the tightened filaments pull the thin filaments outward to their original relaxed position. Due to many contraction cycles, the actin and myosin filaments will slide along one another to shorten the sarcomere. When no longer stimulated, the sarcomeres will return to their previous length.
I guess that's it. That's the important stuff. It's kind of neat to see myosin climbing up actin, right? So many things happen on such a minute scale, and we take it for granted that when you're contracting a biceps, flexing the elbow, how many times are cross bridges being formed and released and formed and released, and that cycle is repeating to allow full movement through the range of motion? Hundreds of thousands of times. That's pretty impressive. So, for myosin to contract the muscle, we need three things. And I didn't write this down, but it reaffirms what was already said in the video. You need calcium, right? Calcium is released when the signal is sent. If calcium's not there, contraction can't happen. Second thing you need is energy, ATP. If ATP is not there, you're dead. But ATP needs to be there for myosin to reach and then contract the muscle. We need energy from some source. And the third point is important for the next slide. The third thing you need for contraction, for myosin to contract the muscle, is it has to have actin to grab onto. Calcium, ATP, and then myosin has to be able to grab actin. If there's no actin to grab, there's no contraction possible. And that last point is really the basis of, or hints at, the length-tension relationship in muscle. The length-tension relationship in muscle. That is, depending on how much overlap there is, or how long the muscle fiber is, how much it's stretched, you can generate a different amount of force. The force that you can generate depends on the overlap of actin and myosin. Maybe think about the amount of force you can generate depends on how many cross bridges you can make at once, how many power strokes you can have occur. There's only a couple, you're not going to generate a lot of force. There's no power. If there are many, you can get a forceful, powerful contraction. So the force of a muscle depends on the overlap of actin and myosin. Somewhere in the middle, there is an optimal range. There is enough overlap. What about outside of that optimal range? Well, if the muscle is too stretched over on the right-hand side, theoretically, there could be no overlap. Sure, maybe there's calcium, maybe there's energy, myosin is reaching, there's nothing to grab onto. There's nothing to grab onto, you can't make much tension, you can't generate much force. If the muscle is too stretched, if there's not enough overlap, force is near zero. As you move towards the middle of this range and there's some optimal overlap, we have the maximum force possible. But then, as the muscle shortens too much, force drops off again. I've said this point already. Tension depends on the overlap between actin and myosin. When the muscle gets too short, the force drops off again. Why is that? Because there's no space for extra shortening. That's what I mean by the space between myosin and the Z discs. If there's a lot of overlap, but myosin is crammed right into the ends of the alleyways, right into the end of the Z disc. As soon as it tries to generate a power stroke, it can't move anywhere. There's no extra shortening possible. It can't move anywhere, it can't cycle, it can't generate force, and so tension is near zero. Somewhere in the middle, though, there is just enough overlap. The Goldilocks zone. Overlap is just right. There's enough myosin, enough actin that cross bridges can form. There's enough space that it can shorten, and we generate peak tension. What I'm describing is in one sarcomere, and it's easy to understand in one sarcomere. You have all these filaments, you can zoom in on them, and you can imagine them moving back and forth. But you can take this theory and apply it to the entire muscle. You've got many sarcomeres lined up in sequence. If my biceps muscle is completely extended, most of those sarcomeres are really stretched, and I can't generate very much force down here. If I tried to generate as much force as possible, it would be quite low. Similarly, if I go to the other end of the range of motion, all of my sarcomeres are really crowded. I can't generate much force up here either. But in the middle of the range, maybe I'm lifting a 25-pound dumbbell, right? Generating a ton of force. 25 is not a lot. That was a joke. In the middle of the range, you generate the maximum force possible. You can lift the most weight possible because your sarcomeres have their optimal overlap in the middle of the range of motion. All right, so what we've described so far is the recipe for muscle contraction. Nerves send the signal to turn on. Calcium is released. Actin and myosin interact. Myosin contracts, shortens the sarcomere. And that general recipe applies to all skeletal muscle. But not all skeletal muscle is born equal. There are different flavors of skeletal muscle. Do you have this? Yeah? Okay. Usually when there's a lot of rumbling and groaning, it's that I missed something in the notes. There are different flavors of skeletal muscle. Maybe that's the comment. You're not actually eating skeletal muscle. Maybe you are. Burgers are skeletal muscle. There are different types of skeletal muscle. They all follow the same general recipe, but they do it in a slightly different way. In humans, really in most animals, there are 3 different types of skeletal muscle. It ends up being a spectrum, but we can broadly classify them in 3 bins. The first type, type 1, slow oxidative muscle. This is muscle used for endurance type activities. You can use it for a long time. It's aerobic, it uses oxygen, doesn't really generate a ton of force, but it's a marathon muscle. You don't need it to generate a lot of force. Slow oxidative muscle is type 1. The others are type 2. There's type 2A and type 2X. On the extreme, type 2X is the exact opposite of what I just described. Type 2X is not marathon muscle. It doesn't use oxygen at all. You can only use it for seconds, but it generates a ton of force. And then it very quickly fatigues. Then you have the best of both worlds muscle fibers. type 2A is somewhere in the middle. It generates lots of force, but it also is fairly aerobic. It's a 50-50 split. I like to think of hockey players as having a lot of this muscle in the middle. It's very aerobic. There's a lot of explosive type activities. The best of both worlds. So these three general bins, type 1 or slow oxidative, type 2A, also called fast oxidative glycolytic. What does that mean? Type 2X, fast glycolytic. The word glycolytic and oxidative, they refer to the type of energy used. We're going to save those words till we get to metabolism. That's coming up after this. What makes these fibers different? Why are there three types? Why are there three types? Well, I've color-coded these boxes according to what they look like. If you were to take a muscle and separate all the fibers out, they would look different. Type 1 would look red. Type 2X would look lighter or whitish. What makes them look different? They look different because they have a different amount of myoglobin in them. Lots of myoglobin in type 1, very little myoglobin in type 2X. And myoglobin is a protein that binds oxygen. It tells you how much oxygen this muscle will use. So type 1 uses a lot of oxygen, and then progressively less as you go through the different types. Why is oxygen important? Oxygen itself isn't important, but it's used in the energy generation process. We referred briefly to mitochondria last class, Dr. Kane's favorite organelle, the powerhouse of the cell. Mitochondria are where oxygen is reduced to water while making ATP. So we'll see that when we come back to metabolism again, but it would make sense if one muscle fiber had a lot of myoglobin, meaning it had a lot of oxygen, it would have a lot of mitochondria. And it does. Type 1 muscle has all of those things. Progressively less and less as you move through the ranks. So the amount of oxygen describes the endurance of the muscle. The other aspect that I mentioned was the force that can be generated. The force that can be generated all depends on what type of myosin you have. Myosin binds ATP and reaches, lets go of the breakdown products, makes the power stroke. How powerful that stroke is depends on what type of myosin you have. There's 6 or 7 different types of myosin protein. Some of them are more powerful than others. They contract more quickly than others. And so the type 2X fibers have really fast myosin. Type 1 fibers have really slow myosin. And then there are really mixtures in between the entire length of the spectrum, but these are the general categories that we can separate muscle tissue into. In every muscle fiber, what we've described up until this point holds true. They're turned on by nerves, calcium is released, and those 4 steps in the contraction cycle occur. They just occur more quickly or more forcefully in some fibers versus others. Let's talk about that idea of force. Force is what you measure externally. So if I were to do a biceps curl and then I measured how much weight I was lifting or force I was producing, I'd be measuring that externally. I'd be lifting against a cable that was attached to a meter somewhere that told me how much force I was generating. Force is measured externally. Tension is what's created inside the muscle. Tension is the pull inside the muscle. and that pull creates the force that we're able to measure externally. So I often use those interchangeably, but they are somewhat different. Tension is generated in the muscle, force is measured externally, outside the muscle. I can do a light contraction, I can do a heavy contraction, I can do a maximal contraction. How? I thought if a nerve just sent an action potential, it was all or nothing. And the muscle would turn on and I would just contract really forcefully. How is it that I can contract partially, for lack of a better word? How can I do a light contraction, a medium contraction, a heavy contraction? How do I control muscle tension in another way? So I'm going to assume that everything is available for contraction to occur. There's enough overlap, there's enough oxygen, there's calcium, there's ATP. The two ways that you can increase tension in the muscle are: one, turn on more muscle; two, turn on existing muscle more. Amplify it, turn it up. So to make a stronger contraction, I can either get more muscle involved, or I can just take whatever muscle is active and I can make it more active. I can amplify the signal. There will be parallels in this next section to temporal and spatial summation. So let's go through these each in turn. One, how do I recruit more muscle? For that matter, how is muscle recruited to begin with? We've talked about a nerve sending a signal and muscle turning on, and that's true. More precisely, muscle is activated in what are called motor units. Motor units. The strength of contraction depends on how many motor units are turned on. How many motor units are turned on? And a motor unit is simply whatever motor nerve and the attached muscle fibers are connected together. One motor nerve and all of the muscle fibers that it ends up innervating is one motor unit. And you can have multiple motor units. Your biceps muscle will have multiple motor units. Every muscle has multiple motor units. A motor unit consists of a somatic motor neuron that you can activate voluntarily and all of the muscle fibers that it innervates. Importantly, all of the fibers in one motor unit are the same. They're either all type 1, or they're all type 2X, or they're all type 2A. All of the fibers that are activated by a motor nerve are the same type of fiber. And so you can see that over in the example on the right-hand side. I've got, let's say this motor neuron 1. You can connect the colors. Motor neuron 1 is this dark bluish green, and it only goes to the bluish green muscle fibers. Motor neuron 2 is the red nerve, and it only goes to red muscle fibers. They're all of the same type. They're either all slow muscle fibers, or they're all fast muscle fibers, or they're all type 2A, what I'm calling a hybrid type of muscle fiber. So when I start my contraction, if I want a light contraction, maybe I only turn on one of these motor neurons. That activates 3 out of the 9 or 10 fibers on the slide. So I create a force that's a third of my potential force. If I activate one motor unit, I only activate some of the fibers in the muscle, and I can create a light contraction. If I only activate one or a few motor units, I'll create a weak contraction. If I decide instead to turn all of them on, I can create a strong contraction. Activate more muscle. So how do I control muscle tension? How do I create a stronger contraction? I can add more and more motor units and create more force. And the body has a really interesting way of separating the motor units out. There's a hierarchy. When you go to the gym and exercise, when you're out running or cycling, the motor units are turned on in a specific order. And that order is what's called Henneman's size principle. Henneman's size principle. Namely, whenever you turn a muscle on, small fibers get turned on first. Only when you need to do you activate medium fibers and then large fibers. Small muscle fibers are always turned on first. It just so happens small muscle fibers are type 1. Type 1, slow oxidative, always turn on first. The hybrid muscle fibers are medium. The fast glycolytic muscle fibers are large. These only turn on in the rarest, most extreme circumstances. Think really heavy weightlifting kind of circumstances. So when you turn on a muscle, you always turn on slow oxidative first. If those can't do the job, you add the medium fibers. If those combined can't do the job, you add the heavy fibers, call in the big guns to get more force. So there's a sequence to how these are recruited. So if you need a lot of force, everything turns on. If you need a little bit of force, only slow fibers turn on. What's kind of interesting to think about is something like running or cycling. You're doing these really light, repetitive contractions, but you're doing them in some cases for an hour or 2 hours. How is it that those muscle fibers are able to allow you to exercise for 2 hours? The answer is in that you share the load. Each muscle fiber has separate motor units, and it has multiple slow motor units, multiple hybrid motor units, multiple fast glycolytic motor units. So as you're running, you might turn on slow motor unit 1 for the first little bit. When that gets tired, you'll activate slow motor unit 2, slow motor unit 3. These will alternate to allow exercise to persist for long periods of time. Motor units of the same type will alternate to allow them not to be fatigued, to allow exercise to persist for a long period of time.